Welcome everybody to the afternoon session. My name is Dan Bernstein, and it is a pleasure to be joined by Natalia Gomez-Espina and Melissa Mavers in moderating this, what I think is one of the most exciting parts of this entire program, the early career investigators and trainees. Indeed, the support, as there was a slide up there about the massive amount of support that the Maternal and Child Health Research Institute has been providing for our early career investigators and trainees. There are a couple of new programs. One is a K support program that we're very excited about, so we're very much looking forward to enhancing the amount of support we can provide for investigators. So without further ado, I will then turn this over to Dr. Gomez-Espina, who will introduce our first speaker.
So our first speaker is Margot Bowen. She's a postdoctoral fellow in radiation oncology, and she is one of the recipients of one of the postdoctoral fellowships.
Thank you. A number of genetic syndromes have been associated with mutations in genes involved in fundamental cellular processes, such as ribosome biogenesis, DNA repair, central duplication, RNA processing, transcriptional regulation, and telomere maintenance. And while each of these syndromes shown here is caused by mutations in different genes, emerging data indicate that a common theme shared by each of these syndromes is that they are associated with activation of the p53 protein. Now, p53 is a transcription factor, most well known for its role as a tumor suppressor. And under normal conditions, p53 is degraded and kept at low levels in the cell. But in response to various types of stress, p53 can become stabilized and activated and induce target genes involved in downstream responses such as apoptosis and cell cycle arrest. And with regards to the syndrome shown here, a number of studies indicate that mutations in these genes can trigger various types of stress responses, such as ribosomal stress, DNA damage response, or mitotic delays that trigger a mitotic surveillance pathway. And all of these different types of stresses can end up activating the p53 protein. And what's really interesting is that many of the developmental defects in mouse models of these syndromes can be rescued by loss of p53. As an example, the craniofacial defects in a mouse model of Treacher-Collins syndrome can be completely rescued by deleting p53. Now, these studies suggest that activation of p53 itself plays a direct role in contributing to at least some of the developmental phenotypes associated with each of these syndromes. And the overall goal of my research is to further understand the role that p53 plays in promoting developmental defects in this diverse group of genetic syndromes. To study the role of p53 in developmental defects, I wanted to use mouse models where I could specifically activate p53 and then study the developmental defects that arose. The genetic tool that I used to activate p53 was to express a particular mutant version of p53 that is able to form tetramers with wild-type p53, but these tetramers are more stable due to the inability of this mutant protein to bind to the main negative regulator of p53. This leads to an overall increase in the stability and activity of p53. And using this mouse model of p53 hyperactivation, my first question was to identify the cell populations in which p53 acts to drive different developmental defects. I made use of a panel of MCHRI drivers which would allow me to express the mutant p53 allele, thereby activating p53 in specific cell types during embryogenesis. Using this approach, I was able to identify a few key cell compartments in which p53 can act to drive different sets of developmental defects. For example, I found that when I activated p53 in a mesodermal population called the anterior heart field, it triggered certain types of congenital heart defects, such as the heart outflow tract defect shown here. I found that activating p53 in endothelial cells could also trigger certain heart defects, such as ventricular septal defects. I found that activating p53 in the neural crest was sufficient to induce a host of phenotypes, including cardiovascular defects, pigmentation defects, and craniofacial defects, such as the cleft palate and outer ear defects shown here. And finally, I found that activating p53 in the facial ectoderm could also trigger a distinct set of craniofacial phenotypes involving the eyes, nasal airway, and inner ear. Together, these studies indicate that p53 can act in multiple cell compartments to drive distinct sets of developmental defects. So now that I had identified the specific cell populations in which p53 can act to drive developmental defects, I next wanted to ask whether p53 needs to be activated at a specific time point within these cell populations. I found that the timing of p53 activation was very important in determining the phenotypic outcome. For example, some of the developmental defects that arose when I activated p53 during the earliest stage of neural crest development no longer manifest when I activated p53 at just a very slightly later developmental stage. These studies suggest that neural crest cells are most sensitive to p53 activation only during the very earliest developmental stage. My next question was to ask whether the exact degree of p53 activation was important in determining the phenotypic outcome. I made use of some additional genetic tools that I won't have time to explain in detail, but they allowed me to trigger different degrees of p53 activation. And I found that the degree of activation was very important in determining the phenotypic outcome. For example, when I activated p53 in the facial ectoderm, I found that a very specific eye defect called retinal coloboma only manifest when I activated p53 at moderate levels, but not when I activated p53 at a slightly lower level or a slightly higher level. These results indicate that p53 must be activated at just the right levels for certain specific developmental phenotypes to manifest. So now that I had identified where, when, and how much p53 activity is needed for different developmental defects, I next wanted to explore the underlying mechanisms by which p53 is able to promote these phenotypes. I started off with some RNA-seq experiments, and I found that activation of p53 in the developing embryo leads to increased expression of a large number of genes involved in apoptosis and cell cycle arrest. I therefore chose to quantify the level of apoptosis and proliferation in my embryos with p53 activation. And I got some very interesting results. For example, when I activated p53 in neural crest cells, I found that p53 activation triggered apoptosis without having any effect on cell proliferation. And interestingly, this apoptosis only occurred during the earliest stage of neural crest development. Furthermore, when I activated p53 in the anterior heart field, I also found that it triggered apoptosis in the progenitor population of the anterior heart field, but restrained proliferation rather than triggering apoptosis in descendants of the anterior heart field that had migrated into and populated the heart itself. These studies suggest that activation of p53 may drive developmental defects by triggering apoptosis or restraining proliferation in only certain cell types and at specific developmental time points. In summary, I identified 4 key cell populations in which p53 can act to drive different sets of developmental defects. I found that the timing of p53 activation and the degree of p53 activation is critical in determining the phenotypic outcome. And finally, I showed that these developmental defects are associated with increased apoptosis or decreased proliferation in specific cell types at specific developmental time points. And bringing it back to these human syndromes, my data indicate that activation of p53 alone is indeed sufficient to induce many of the developmental phenotypes observed in these human syndromes, thereby supporting the notion that activation of p53 could be a key mediator in these syndromes. And secondly, my data indicate that phenotypic differences between these syndromes could arise due to even slight differences in the exact degree of p53 activation or the exact time point at which p53 becomes activated in these syndromes. And further research will be needed to understand how the different causative mutations in each of these syndromes is able to trigger p53 activation to different degrees or at different time points. And with that, I would like to thank everyone in the Attardi Lab for their help with this project, and I would like to thank the CHRI for providing funding and for giving me the opportunity to speak here today. Thank you.
Questions?
So I'll start, if I'm allowed to. And a very elegant work, and actually a lot of things to follow up on. But a lot of the effects of p53 may or may not be mediated through cell-autonomous effects. And I wonder if you have plans to look at the interaction between the cells that you're expressing your mutant p53 and other cell types. And the example in the heart would be the effects of the heart on neovascularization, development of developing vasculature in the developing heart.
Yes, so when I created my mice with activation of p53 in different cell types, I analyzed a lot of different phenotypes, and in general, I mostly only saw phenotypes arising through to cell-autonomous defects. For example, when I activated p53 in the facial ectoderm, the ectodermal tissue interacts with neural crest during development, so it's possible that you could get neural crest defects. But I, for example, did not see heart defects in those embryos, suggesting that the cardiac neural crest was at least forming properly. But there certainly could be some phenotypes that arise through to interactions with different tissues.